let's talk about something that drives every lab scientist crazy. We're going to dive into the hidden forces that can absolutely wreck a 96-well plate experiment. And over the next few minutes, I'll show you how to stop being a victim of your data and start becoming its architect. Let's get right into it.
You know the feeling, right? It's that specific kind of lab heartbreak. You pull your data, you're expecting this beautiful clean curve, and instead you get this, a total mess. Hot spots, weird smile patterns, drifting values all over the place. It's so frustrating, and man, it really feels like you failed.
And your mind just starts spinning, doesn't it? Is this antibody lot bad? Did I mess up pipetting row G? I was probably tired. You start questioning everything, your technique, your tools, maybe even your own skills, but what if? What if the problem isn't you at all?
Yeah, you heard me. It's physics. More often than not, the real culprit isn't some random mistake you made. It's systematic bias. It's the predictable physical forces that are acting on your plate every single time. And here's the best part. Once you understand these forces, you can design your experiment to make them practically disappear.
Okay, so let's meet these hidden enemies of good data. There are really three main culprits we need to drag into the light before we can figure out how to beat them. We're talking about plate effects, timing drift, and batching.
So, first up, plate effects. You gotta stop thinking of a 96-well plate as a perfectly uniform grid. It's not. It's more like a tiny landscape with its own microclimate. The outer wells, they're exposed to more air, which means faster evaporation and different heating rates. This is the infamous edge effect, and it can seriously skew your results before you even get to the reader.
Next on the list is timing drift. In immunoassays, time is basically a reagent, but it's a reagent you can never measure out perfectly. Think about it. If it takes you just 5 minutes to load a plate, well, the very first well has had 300 more seconds of reaction time than the very last one. That little head start, it can create a huge signal gradient that has absolutely nothing to do with biology.
And finally, we have the silent killer, batching. This is something we all do, right? We group samples because it's convenient. We run all the healthy controls on Monday and all the patient samples on Tuesday. Or maybe one tech runs the morning plates and another one does the afternoon batch. It seems efficient, but what you're actually doing is making it impossible to tell if your results are a real biological effect or just the Tuesday effect.
So now that we've met the enemies, how do we fight back? Well, the answer isn't about working late nights or buying some fancy new robot. Nope. The single most powerful solution is something called run order.
That's the secret weapon right there. It's not about the expensive stuff, it's about being smarter with your setup. It's all about how you strategically place your samples on the plate, that's the space part, and how you process them, that's the time part. Get that right, and you'll separate the real signal from all that noise.
So let's open up the toolkit. I'm going to walk you through 4 core principles that will literally transform you from a simple kit user into a true assay architect. Someone who can build a completely bulletproof experimental design. All right, here's the playbook. 4 simple steps. First, block your design. Just means you make sure every single plate has a balanced mix of all your sample groups. Second, use what's called restricted randomization. You're mixing things up, but in a way that prevents all your controls from clumping in one corner. Third, protect your standard curve. Get it away from those sketchy edges and into the stable center of the plate. And fourth, and this one's huge, interleave your replicates. Let's look a little closer at why that one is such a game changer.
Okay, look at this. It just shows you the power of smart design. On the left, that's what most people do, replicates right next to each other in A1 and A2. Well, what happens if a little bubble or a splatter hits that corner? Boom, both of your data points are garbage. But look at the right. The replicates are placed far apart. Now, if that same local error happens, it only takes out one of them. Your average is suddenly way more reliable because you've literally averaged out the physical weirdness across the entire plate.
Now, this all sounds pretty good in theory, right? But does it actually work in a real lab under pressure? Oh yeah. Let's look at a case study that shows just how dramatic the impact of this can be.
So, get this. A lab was in a really tough spot. They were struggling with this critical IL-6 ELISA. Their coefficient of variation, their CV, was over 25%. I mean, that's just incredibly noisy, totally unreliable data. They couldn't replicate anything and were literally about to drop 20 grand on switching vendors because they were sure the reagents were the problem.
But hold on, they didn't change the reagents, they just changed the design. And just look at this result. By simply rearranging how they ran the exact same samples with the exact same reagents, their CVs plummeted from a crazy 25% all the way down to a beautiful, clean 6%. That signal they were looking for, it was there the whole time, just buried under all that noise.
So what was the secret sauce? This is basically the cheat sheet right here. They made 3 dead simple changes. 1. They moved the standard curve from the sketchy edge column into the stable center. 2. They stopped running samples one group at a time and started interleaving and randomizing them. And 3. They stopped putting their replicates side by side and spread them far apart. That is it. No new equipment, no new kits, just smart thinking.
Okay, so let's make this super practical for you. Here is a simple pre-flight checklist. Just ask yourself these 5 questions before you run your next assay, and you'll make sure your design is working for you, not against you.
Let's run through it real quick. Number 1, are my groups balanced on every plate? 2, where's my standard curve? Is it safe and sound in the middle? 3, are my replicates separated to average out that spatial noise? 4, how am I handling the clock? Am I keeping my timing and rhythm consistent? And number 5, this is for you long-term study folks, do I have an anchor, like a bridge sample, to tie all my plates together over time?
You know, in the end, it really just comes down to this one question. Anybody can follow a kit insert. That makes you a user. But understanding the physics of your plate and designing your run order to conquer bias, that makes you an architect. You stop fighting your data and you start trusting it. So for your next experiment, which one are you going to be?